Hello, I'm David Field, and this is the PYM0FM Brain Imaging Using FSL MSc course. I'd like to thank the SIN technician Jason Hill and the Oxford fMRI or fMRI, which stands for Functional MRI of the Brain Laboratory. And they're the ones that create the FSL software, and I'm using some of their materials in the course. Now let's begin with this image of a brain on a plate. So that's quite amusing. We don't often eat brains, and if we do, they don't look like that. They're usually in a soup or something. But the reason I've put that image there is because the brain itself is very hungry. It actually uses 20% of all the energy that we consume. That's 20% of the glucose that gets into our bloodstream from food we eat is consumed by the brain. And that's a really high percentage, considering that it only weighs or only accounts for 2% of our body mass. Now, of course, what we're doing in fMRI is imaging the energy that it's using, or at least imaging the oxygen that it's combining with that glucose in order to get an idea of what the neurons are doing. And this second image is just a picture of the vasculature inside the brain that delivers all that nice glucose and oxygen to the neurons and removes the waste products such as carbon dioxide in the blood. And that image is important because that's what we're actually imaging, really, when we're using fMRI. We're imaging something to do with the blood supply system to the brain, right down to the small capillaries, but also including some of the larger blood vessels. Okay, so on this slide I'll just give you an overview of what we're going to do in the first session. The workshop or practical session is going to cover using the FSL software, which is actually made up of a number of small sub-programs, to view brain images. You know, imaging is a very visual thing. You've got to be familiar with how to navigate around brain images and know what you're looking at. And the program that's provided for doing that is called FSL View. Also in the workshop we're going to cover the program BET, which does a process called brain extraction. Brain extraction is just removing the skull and cerebrospinal fluid parts of the image in order to isolate the brain. And it's just important to do that in order that those things then don't get in the way of the later functional analysis. The somewhat amusingly named FLIRT program performs a number of useful functions. It does something called intra-subject registration. Now, intra-subject registration is what you do when you've got images from the same person, but those images have got differences. So, for example, one of them might be an image from a PET scanner of that person, and another one might be an image from an fMRI scanner. So then you'd use FLIRT to match up those two images to make a point-by-point correspondence between them. Another example would be if you've got the high-resolution anatomical image from the MRI scanner and you want to make a point-by-point mapping of that with the low-resolution functional data from the same MRI scanner and the same person. Inter-subject registration with FLIRT is just an extension of that to when you want to make kind of point-by-point mappings between different participants, so that you can sort of say, well, this is for example the superior colliculus in both people. So that's achieved via registering both of those individual people to a standard or template space. Much more about that later. So in this lecture, which is really the preparation for the workshop, I want to try and give you the practical concepts that you'll need just to be able to understand what these various bits of software are doing, but also I want to give a theoretical and background and overview of how an MRI scanner works and how it produces the images that you're going to be analysing in this course. And also at this stage, before we move on, I'd just like to apologise for my cold and sorry for the effect it's had on my voice. So I'm going to begin by considering the problem of imaging in a very general sense. As a methodology, what is imaging? Then we will consider what is imaged in MRI. That's magnetic resonance imaging, and it's exactly the same thing they use at the hospital to get anatomical images of parts of people's bodies. So there's nothing functional about this, it's anatomical, but it's got a lot in common with the functional stuff, so we will consider it in some detail. And of course anatomical images are also used in psychological research. So we need to consider later in this lecture the coordinate system that we use to navigate around the images and allow us to communicate with other people about what part of the image we're talking about. We need to define the concept of a voxel, which is very important in MRI. Look at the underlying physics. Now, I'm not a physicist, so there won't be any equations, but we'll try and get an idea of how the MRI scanner works, which is basically what it does to produce these image intensity values. And we need to consider two types of contrast, T1 contrast and T2 contrast. Then we'll look at bit more briefly at what's imaged in functional MRI. The physics of this involve looking at something called T2 star contrast. And then we'll move on to BOLD, which is stands for blood oxygen level dependent. And here we start to cross over from the physics into the physiology. So there we're looking at the hemodynamic response. Hemodynamics refers to blood flow, in specifically the hemodynamic response to neural activity. And we'll introduce the concept of a time series of data in which there are three spatial dimensions plus the fourth dimension of time. And then we'll also briefly today have a look at the steps in the analysis of fMRI images. This is a topic we'll cover in more detail in later weeks of the course, but these topics are motion correction, registration, affine transformations, specifically for registration of motion correction, brain extraction, and how we model the time course. So, what is imaging and when do we use it? If we're interested in quantity A to answer our scientific question, but we don't have a way of measuring quantity A, we may often find that we're able to measure another quantity B which has some kind of systematic relationship with quantity A. So we're using measurement of B to infer something about the unobservable quantity A. And in order to do this, we need some sort of model of what the relationship or coupling is between A and B. It may not be a simple linear relationship of the sort that you may be used to considering in, for example, multiple regression. Now, let's use an example. It's an artificial example or an arbitrary example, but it has properties that are true of all imaging systems. Imagine it's a sunny day, that'd be nice, wouldn't it? And you're standing near a wall, and the sun is shining on the wall, and you can place sets of different types of objects between the sun and the wall. You might put a solid object like a football. You could hang a shirt there between the sun and the wall. You could hold up some sunglasses. You could produce a cloud of water vapour. perhaps by boiling a kettle. Or you could put a pane of glass between the sun and the wall. And then let's say we've got some hypothetical scientists, and all they've got is a photometer, which is able to measure the brightness of the light being reflected off the wall, i.e. they can measure, in a sense, how much shadow is being cast by each of these objects on the wall, and they could also measure the light being reflected from the part of the wall where there's nothing interposed between the sun and the wall. So they get their they measure the amount of light reflected when nothing is interposed, and they can then measure the amount of light reflected for the solid object, for the shirt, for the sunglasses. And obviously there'll be much less light reflected off the wall in the case of the solid object than there will for objects that allow light to pass through, like shirts and sunglasses. But then water vapour perhaps blocks even less of the light, and the pane of glass blocks very little of the light. So what we could actually infer with this simple imaging system is something about how opaque the objects are, just by using a photometer to measure the shadows and tell us about the density of the objects. But this imaging system is limited. We would not be able to measure the object sizes by measuring the size of the shadow, for the simple reason that the size of the shadow depends not just on the size of the object, but also on the distance of the object from the wall. The lesson there is that all imaging techniques tend to be quite limited. They're only able to measure highly specific things, and when you try to infer too much using them, you often get into uncertain territory. Now, in the case of functional brain imaging, the actual coupling or relationship between what you measure and the thing you're interested in, which is a neural activity, is a very indirect and multi-stage story. You're normally interested in some psychologically relevant stimulus, actually, and then you get the neural response to that. There is some vascular response to the neural response, and then the MRI signal is influenced by the vascular response. So clearly the model of this relationship or coupling is going to be complex. Now let's take a look at what is imaged in MRI specifically. These are images that, of course, are intended to provide information mainly about anatomical structure. In other words, contrast is required between different tissue types in order to distinguish how tissue type varies over space. These, of course, are used in hospitals and don't contain any information about variation over the time course of the scan. And when you're doing functional brain imaging, you also acquire the anatomical scans in addition to your functional scans, because your functional scans contain only relatively coarse information about anatomy. You want to also make use of MRI images that contain good information about the anatomy of your participants, and then you'll be able to co-register the two types of image, which we'll see in the workshop. So we'll begin by taking a look on the next slide at the end product, which is, in terms of our sort of description of imaging, it's the imaged quantity. And then in order to understand it better, we'll take a look at what the actual directly measured quantity is in anatomical MRI. So these images on the left here are an anatomical image from an MRI scanner. It's called a T1 weighted image. More about that later. And in these images, cerebrospinal fluid appears very dark. Bone tends to appear as white. Now, your white matter tends to appear as lighter in colour than your grey matter, and that's because it has a higher fat content. So now I'm kind of pointing at some grey matter, I don't know if you can see, and in the middle here is the white matter. Now in another type of image called a T2 image, that contrast of the grey matter and the white matter is reversed. So then you'll actually see that the white matter is darker in the image than the grey matter, and the T2 images are often the ones they use in hospitals to reveal pathologies such as tumours. So just a little bit of stuff about terminology now. If you see an image like this, which has kind of been sliced as if you're slicing from one ear to the other of the brain, of the head, that's a coronal image. If you see an image that looks like it's been sliced from the nose to the back of the head, that's a sagittal image. And if you see an image that looks like it's been sliced from the front of the head to the back of the head, then that's an axial or transverse image. Now these T1 images are generally actually acquired by the scanner as sagittal slices. It's actually doing the acquisition by slicing through the brain in a series of sagittal slices, and then the coronal and axial views are subsequently reconstructed afterwards by the computer. Okay, so I just wanted to show you this slide because this is as good as it gets. This is a T1 image that exhibits exceptionally good grey matter to white matter contrast. You can see that I'm able to zoom the image in a lot more because there's a lot more native spatial resolution in the image, and you can see that I'm tracing with my red dot around the white matter really easily here, and you can see it's really easy to see the difference between the white matter and the grey matter. Now this was acquired in the scanner using a sequence called an MDEFT, in contrast to the image on the previous slide, which was I think an MP-RAGE standard sequence. The disadvantage of this is it takes 20 minutes to obtain this image versus 5 minutes for the standard MP-RAGE, but if your research interest requires you to segment the brain into separate white and grey matter images, perhaps because you'd then like to computationally flatten the cortical sheet, then this is the type of image that you really need in order to make that process work well. You've probably seen some journal articles where they publish flat maps of the brain instead of brains as we're used to seeing them, because of course with a flat map they've unfolded all the gyri and sulci, and you can see much better in those valleys and fissures. And if they've done their research thoroughly, then they've probably begun with a sequence like this one in the scanner and paid the cost in time to get these high quality images. On this slide we have a little bit more terminology for you concerning directions and locations. The top of the brain is referred to as dorsal, whereas lower down in the brain is referred to as ventral. And these can be used both as locations, such as the dorsal part of the visual system, which is up here, compared to the ventral part of the visual system, which is down here. And they can also be used as relative locations. So you can say that brain area A is dorsal to brain area B. And of course the fact that it's dorsal to brain area B, it may itself be ventral to another brain area. Another important terminology is anterior and posterior. Anterior just refers to the front of the brain, and posterior to the back of the brain. You will also hear the term rostral to mean anterior, and the term caudal to mean posterior. Things near the midline of the brain, this is the midline I'm highlighting here, are referred to as medial, and things near the edge of the brain are referred to as lateral. Here I'm just showing medial and lateral as they would be appearing on this coronal slice. And of course these things, as I said, concerning dorsal and ventral, these could also be used as relative locations as well as kind of absolute locations. Now, as well as having a set of words that we can use to sort of roughly refer to locations and directions in the brain, we also need a numeric coordinate system, and this is referred to as the stereotaxic coordinate system, although it's also referred to as the Talairach coordinate system, and I'll explain why that is, and it's for historical reasons, in a minute. So the basic system is common to all brain imaging studies, and you'll find this then in all the journal articles, is this XYZ system, where a positive value of X indicates that you're on the right hand side of the brain, and a negative value of X indicates that you're on the left hand side of the brain. I always get those mixed up myself, just like I used to get my left and right hands mixed up until I was about 11. So positive values of Y are anterior, front of the brain. Negative values of Y are posterior, back of the brain, like the visual system and stuff is there. Negative values of Z are the bottom of the brain and the cerebellum under the brain. Positive values of Z near the top of the brain. Now, so therefore each voxel in the brain, I haven't defined the word voxel yet, but you kind of know, I expect already, that it's like a pixel, but three-dimensional. Each voxel has a location defined by a value of X, a value of Y, and a value of Z. So the reason it's sometimes called the Talairach coordinate system is because it was first applied to a particular template brain that was the post-mortem brain of, I think, an alcoholic 70-year-old woman. But this was one of the first popular template brains, to which brains of participants in MRI studies are then registered or matched to. Now, so within the Talairach template brain, they used this coordinate system, so it became known as the Talairach coordinate system. But things have moved on, and we now have better template brains, such as the MNI template brain, which is a much more sort of scientific approach to having a template brain, because it's based on the averages, the average brain made across quite a few individuals, and they still use the same coordinate system for the MNI brain, i.e. they use the stereotaxic or Talairach coordinate system, but within a better template brain. So basically, don't confuse the coordinate system with the space that it's in, the brain, the template brain that it's in. Now, this confusion that does exist is actually present in the published literature. Often when you read, particularly the older functional brain imaging papers, it's hard to tell which of the template brains they've used. So do look out for that one, because they are slightly different shape. Now, what I didn't tell you on the previous slide was, of course, the origin of the coordinate system. Any numeric coordinate system only makes any sense at all if it has an origin, that being the point where all values are 0. So x equals 0, y equals 0, z equals 0, the centre of the cube, if you like. So for the Talairach or stereotaxic coordinate system, the centre of the coordinate system is at the anterior commissure, which is shown as a red dot here in this schematic of the brain. And now what you need to do in order to get your origin on there is also to make the y-axis lie on a straight line that is drawn between the anterior commissure and the posterior commissure, and that defines the origin of the coordinate system. Now, when I began doing brain imaging myself, I used the SPM software, quite an old version of it, and in that software you had to look at the T1 anatomical image of your participant and manually set where you thought the origin was, i.e. put it on their anterior commissure by clicking, whereas using the modern version of FSL, anyway, the thing you're going to use, it automatically uses image processing to set the origin for you, which is fine, but I actually think it was quite nice to have that kind of interaction with your data and for you to sort of get to know really where the origin is by having to set it yourself. So perhaps you could have a look at some images to see if you can learn to identify the anterior commissure. So just to help you with that, if you want to do it, this slide shows you what it looks like on a kind of typical MP-RAGE structural image. You can just kind of see it there. It's probably the best view of it, right? That's the origin of the coordinate system. Now let's define the important term voxel. We've already seen on the previous few slides that at each unique combination of x, y and z coordinates you find one voxel. Voxel stands for volumetric pixel, and the brain in your image is kind of made up of a lot of these small square pixels all kind of stacked together to produce one cubic image, because we've kind of, when we begin at least, got parts of our image that actually correspond to air that was sort of outside the head. So therefore we've got a kind of cubic image when we first take our images off the scanner. So we've looked at the kind of address or location of a voxel. What information does the voxel contain about the tissue at its address? Well, it's just got one number, and that one number is a value of image intensity. That's all it is. And of course this single number, this value of image intensity, is useful to us because grey matter tends to have image intensity values that fall within a certain range, and white matter tends to have image intensity values that fall into a different range. How big is the space that each voxel occupies in the brain? Well, for the structural images of the sort I've been showing you, the MP-RAGE standard ones, it would typically be a 1 by 1 by 1 voxel, sorry, 1 by 1 by 1 millimeter cube, while in the functional images that we haven't looked at yet, it's generally a larger cube. A very typical number would be 3 millimeters by 3 millimeters by 3 millimeters. And it's important to note that that functional image is not 3 times larger than that 1 by 1 by 1 structural image. It's actually 27 times larger, because the volume of cortex, or volume of whatever tissue is in the voxel, is 1 cubic millimeter in the case of the anatomical or structural image, whereas it's 3 times 3 times 3 equals 27 cubic millimeters in terms of for the functional image. So how do we get to see these things on our computer screen? Well, it's just an arbitrary mapping really of the values of image intensity at each voxel onto a value of brightness on the computer screen. What we're going to do in the next section of the lecture is look at exactly how the scanner generates that value of image intensity that we've got at each voxel location. This image is a block diagram of an MRI scanner. An MRI scanner has 3 main components, and in this section of the lecture we're just going to have a quick tour of how each of those three main components contributes to ultimately making the value of image intensity that you have at each voxel of the image, as we saw on the previous slides. So the three main components are the magnet, the static magnetic field that's turned on all the time, even overnight when there are no participants in the scanner. In the 3 Tesla MRI scanner that we have here in the Centre for Integrative Neuroscience and Neurodynamics at Reading University, that 3T magnet is 50,000 times the strength of the Earth's magnetic field. Now, the next important component is the gradient coil, and you can see that the gradient coil is connected to these 3 amplifiers here, and that each of these amplifiers has a little letter X, Y or Z, and that X, Y or Z might give you a clue about what the function of the gradient coil is, but more about that later. The third component is the radio frequency coil, and this is the sort of birdcage like thing that the participant's head is inside while they're being scanned. So we'll first talk about the role of the static magnetic field. The important thing about this is that it should be homogeneous. The strength of the magnetic field should be roughly the same, at least over the area where you've actually got a bit of the participant you're interested in imaging, because unevenness or inhomogeneity in the magnetic field will produce problems in the images. So what does the static magnetic field actually do to your participant when you put them in there? Well, the key thing to remember is that it influences the hydrogen atoms in the participant's body primarily, and these hydrogen atoms are present because of the large amount of water in the person. Remember that water is H2O, so it's the hydrogen atoms in the water whose protons are influenced by the static magnetic field. And each of these protons has can be characterized as having an orientation and a frequency. And in this diagram here on the left, the orientation of the proton is this angle that I'm highlighting here with the red pointer. Okay, so if it had a bigger angle down there, it would have a different orientation, right? Now, the frequency is how rapidly it's spinning, how many times per second it rotates, and when the proton is in a higher energy state, perhaps because you've heated the person up, it'll be spinning faster. But the key thing to think about on this slide is the orientation. Now normally, as shown in this diagram on the top right here, the orientations or magnetic dipoles are just kind of randomly distributed in different directions, so the person has a net magnetic there's a net zero magnetization. But when you apply a strong external magnetic field, such as that of the scanner, and this magnetic field is often referred to as B0, the patient becomes polarized and a net magnetization appears. And what that means is just that a lot of these protons change their orientation to line up with the orientation of the magnetic field. And because of the fact that the person is lying supine on their back, you can think of this as the orientations of the protons lining up with the z-axis in your image of the brain. Sorry, I got that wrong actually. They're actually lining up with the y-axis in your Talarat coordinate system, aren't they? But it's the z-axis of the magnet. Now let's move on to look at the RF coil. This is the radio frequency coil, and it's the thing like a bird cage that your participant's head is contained within while they're in the scanner. The RF coil has two jobs. The first job it has is to transmit pulses of energy into the participant's head. This energy is absorbed by the participant, and then after it's been absorbed, it's emitted from the participant. And the second job of the radio frequency coil is to register or measure this energy which is emitted back from the participant as current in the radio frequency coil. Now, more specifically, what the radio frequency coil does is to generate an electromagnetic field at the resonant frequency of hydrogen nuclei. Those, remember, are the hydrogen nuclei in the water molecules in your body. Now, if like me and most psychologists, you didn't really study physics beyond the age of 13 or 16, you might not know what the basic physics concept of resonance really is, and it's quite amazing, and it might seem quite magical to those of us who are not really familiar with physics. So we'll take a look at that in detail in a minute. But let's just say for now that this resonance phenomena is so important in MRI, and we know it is because that's what the R is in MRI, right? Magnetic resonance imaging. And the basic idea is that the RF coil transmits the energy into the body at a specific frequency, and that specific frequency is chosen to be a special frequency called the resonant frequency of the hydrogen nuclei. And because it's the resonant frequency of the hydrogen nuclei, they absorb the energy and other molecules in the head don't. Right, so let's just explore resonance a little bit more. As I said, it's a really fundamental concept in physics which is exploited to allow the radio frequency coil to selectively influence the hydrogen atoms. And what property of them that it specifically influences is their orientation. Remember, we saw the orientation on the diagram earlier. Now, in order to understand the concept of resonance, it's really a lot easier if you see some demonstrations. So please watch these YouTube videos. You should be able to just click on the links, but if that doesn't work, or if you're watching a version of this presentation which has been converted to a video file for YouTube, then you can find those links by going to the YouTube website, and for the first one you search for the words conceptual physics demo of resonance. For the second one, type in bridge resonance Bob Barrett, and for the third one type in resonance experiment Brood Strange. The third one's probably really important, and the first couple just show you kind of impressive looking demonstrations. So really, it is really important to go and watch those. Now, the key point is that in order for energy to be transmitted from object A to object B, A has to vibrate at the correct frequency, which is the fundamental frequency of B. And as you will know if you've watched the videos, every object in the world has a fundamental frequency, and if you can find it, you can transmit energy into that object in the form of vibrations. Now, in the case of the radio frequency coil, it's a sensitive device, so you can vary the frequency of the energy which is being emitted from it. and so therefore you can tune it so that your pulse energy would be absorbed by different atoms. And what we usually do in MRI is set the RF pulses to have the fundamental frequency of the hydrogen atoms. But there's a really important caveat to this, which is that actually the fundamental frequency of hydrogen atoms itself can vary. And so that's another thing which is exploited and is part of the story of MRI. So on this slide we'll just explain what the RF coil allows us to do. Basically, when you apply a pulse of radio waves, bear in mind that this is a really short pulse, it might be like 50 or 60 milliseconds at the resonant frequency of the hydrogen nuclei, what happens is you change the orientation of the spins. Remember that they were all lined up with the direction of the static magnetic field, but you can briefly, with a pulse, cause the orientation of them to change, as shown in this diagram here. But once you turn the RF pulse off, the protons begin to relax, and as they relax, they return to their original orientation, which was lined up with B0, or the static magnetic field. And during that process of relaxation, they emit the energy that they absorbed in the form of radio waves. And now the RF coil is in receiver mode, and it measures that energy, and so that energy becomes electrical current in a coil measured in amperes, and that, if you like, is what is the basic dependent variable in MRI, and in fMRI it is electrical current measured in amperes in an RF coil. Now the relaxation time is an important concept. That is how long it takes the protons to return to their original alignment with the static magnetic field after you terminate the RF pulse. And that's going to vary, as we will see, for different tissue types. Now, the other things that we're going to need to be able to do in order to actually get images from this procedure that we can use is to work out where in the participant, i.e. what voxel, the current is coming from, and also to understand how the variation in the amount of current and the duration of the current is related to the tissue types. It's at this point that the gradient coil enters the story. It's the job of the gradient coil to allow us to spatially encode the MRI signal, i.e. tie those values of current in the coil to particular XYZ values in the brain, that is XYZ values of location. So how the gradient coil works is that it's able to modify that homogeneous magnetic field B0 to make it no longer homogeneous. It's able to introduce a gradient into the magnetic field, that is a gradient in the strength of the magnetic field, either in the X direction, in the Y direction, or in the Z direction, which is why we have these three separate amplifiers here. And note that these gradients are not independent magnetic fields, they're modulations of the strength of the static magnetic field. Now, why it's important to be able to do this and why it allows us to actually encode a signal spatially is because of the caveat I mentioned earlier. The resonant frequency of the hydrogen nuclei actually varies as the strength of the magnetic field that they're in. Now, here's an example of how that works and how that can help us. Here, a gradient has been introduced in the magnetic field strength in the z-axis direction of the magnet, which is the y direction in our stereotaxic coordinate system. So because the magnetic field is now stronger up at this end than it is down at this end, we actually then have a higher resonant frequency of our hydrogen nuclei up here than we do for the ones down here. So now we can tightly tune the frequency of the energy pulses being emitted by the RF coil to correspond to the resonant frequencies of these hydrogen nuclei in different locations in the body. So then we know, because we transmit at a certain frequency, where in the body that frequency can be absorbed, but at other points in the body it can't be absorbed. So then when we move the RF coil into receiver mode, it can only be receiving current from a particular location in the body, and that's how we know, in principle at least, where the signal is coming from in the body. Now that section was perhaps a little complicated because the physics of an MRI scanner is fairly complicated, so let's do a quick recap. The static magnetic field, B0, causes protons in hydrogen atoms to become aligned with the magnetic field. The RF coil transmits energy into the participant and receives that energy back, and that energy is the raw MR signal and its variation in electrical current, essentially. The gradient coil has the job of allowing the signal to be assigned to spatial locations, i.e. a voxel. So the image intensity value at each voxel is derived from the amount of electrical current received by the RF coil for that voxel location. Now the reason that any of this is useful is because the amount of current received by the RF coil at each voxel depends on the tissue type, and therefore tells us what tissue type occupies the voxel. And that's the next thing that we want to try and understand. The reason that the amount of current received by the coil is dependent on the tissue type that occupies each voxel is because the relaxation time of hydrogen nuclei varies depending on the tissue that those hydrogen nuclei, i.e. the water, is in. Now, the time for full relaxation to occur, by which I mean that all the energy that was absorbed has been emitted again, the time for that to occur is governed by a rate constant called T1, which varies between tissue types. T1 is a much longer time for the water in cerebrospinal fluid than it is for the water in tissue. And that's why in the end CSF appears black in the structural image, because there was very little T1 signal for it, whereas other tissues appear brighter. Now, the key thing to understanding this is actually to understand why just applying a single RF pulse and then turning on the receiver coil and waiting won't generate the differences between the tissue contrast that we're interested in. I've drawn some fairly crude graphs that I hope can illustrate this point. So on this graph, at time 0 on the horizontal axis, the radio frequency pulse has ended, and then time will pass, and we'll measure on the vertical axis the cumulative energy that's been emitted due to the relaxation of the hydrogen atoms as their orientations return to be lined up with B0. And so if we just turn on the receiver coil after this RF pulse has ended and wait, then this orange tissue type here has a short T1 constant, and this blue tissue type has a longer T1 constant, so it takes longer to emit its energy, it does it more slowly. But with the RF coil on and receiving for all this time, what will happen is that we'll just recover all of the energy that we put in for both tissue types. So the actual measurement will be the same for both of them. This is illustrated by the dotted line that I've drawn across here to meet the y-axis. The actual value of current in the coil would be the same. We would have lost the information that it took longer for one tissue type than the other, because that's not recorded, it's just the total current in the coil. So in practice, how we get around this is through the use of multiple RF pulses emitted in fairly quick succession. Now the start of this graph is the same as before. An RF pulse is finished, and both the tissue type with the short T1 constant in orange and the one with the slower T1 constant in blue start emitting their energy at their different rates. But then, after a short period of time, we switch off the receiver coil and switch on another RF pulse. The effect of the second RF pulse is to stop the energy emission due to relaxation that was happening due to the first RF pulse. And that's because this second RF pulse once again puts the orientations of the hydrogen nuclei into this excited state where they're different from the orientation of the static magnetic field. But then, once this stops, relaxation begins again. And you can see I've repeated this for a third time here. And now, because we terminated relaxation early, if we draw our dotted or dashed lines across to meet the y-axis, we can see that different amounts of energy get received in the coil from the two tissue types. So if orange tissue type was at voxel X10, Y10, Z11, and blue tissue type was next door at X10, Y10, Z9, say, then we would now be able to distinguish them from each other. So just to kind of repeat that, the T1 tissue contrast is realised by applying multiple excitation pulses in quick succession using the RF coil. Tissues with a short T1 rate constant have time for substantial relaxation to occur between pulses and generate high signal in the receiver because they emit a lot of energy quickly. But tissues with long T1 rate constants give lower eventual signal because most of their relaxation or energy emission process doesn't actually have time to occur before the next RF pulse is applied, which stops the emission of energy that was occurring from the previous